wanted to make a quick video about the different macromolecules. So hopefully by now you've been able to watch the 5 videos that are posted on Canvas about the macromolecules. And hopefully out of those videos you were able to determine what the name of the monomer and the bond and what the function for each of those macromolecules is. So let's just take a minute to talk about each of these. We started with carbohydrates. And this, as we discussed, is primarily important for energy storage, and it's actually quick energy storage, so it can quickly be released. So it plays a role in energy storage. And some carbohydrates actually play an important structural role. And for example, we talked a little bit about cellulose, which is found in the cell wall of plants. And we also talked about chitin, which is found in exoskeleton of insects and in the cell wall of fungi. So there's these two important roles, energy storage and structural support. And the third role for carbohydrate is an interesting one. This is in cell identity and communication. And so we talked about the example of blood groups, which can let one cell know the identity of another cell. Okay, so these are the primary functions for carbohydrates. The monomer is called a monosaccharide. That just means one sugar. That's what that word means. And there are multiple different types of monosaccharides. Some examples that you may be familiar with are glucose, fructose, and galactose, just to name a few. Those are examples of monosaccharides. You probably are also familiar with some examples of disaccharides once you put two sugars together. So for example, if you put glucose and fructose together, you get sucrose, which is table sugar. Okay, but that's a disaccharide. So when we take two monosaccharides and put them together, we form a special bond called a glycosidic bond. Glycosidic bond. And that attaches two sugars together. Okay, I'm actually going to add to this. After carbohydrates, we talked about nucleic acids. Okay, the purpose of nucleic acids, the function is to store and transmit genetic information. Okay, so we had talked about the examples of deoxyribonucleic acid and ribonucleic acid as the major nucleic acids. The monomer for each of those is called a nucleotide. And we talked about the fact that there are actually multiple different nucleotides. In DNA we have A, adenine, thymine, cytosine, and guanine. And in RNA we actually have A, U, C, and G. We have uracil instead of thymine. Okay? These individual nucleotides are attached together through phosphodiester bonds. And that basically builds the sugar phosphate backbone. And then we talked about not a covalent bond, but another bond that actually supports DNA structure are the hydrogen bonds that are found between the base pairs. But the covalent bond that forms the polymer is called a phosphodiester bond. Okay, the last one we have here, I'm going to go to a new slide here. The last of the macromolecules was protein. Okay, so protein is built from a monomer called an amino acid. So So the amino acid is the building block, and we mentioned that there are 20 different amino acids that build proteins. These amino acids are tied together through a specific type of a bond called a peptide bond. And remember, I talked to you a little bit about how to recognize that peptide bond, in that if we look at the structure of an amino acid, you always have a central carbon. You always have an amino group on one side. And on the other side, you have a carboxyl group. Oops, let me erase that guy. Okay. You have an R group, and then down here you have a hydrogen. When you form, this is one amino acid, but when you form a peptide bond, I'm actually going to go and draw this on another slide here. Here we go again. Here is the amino acid. When you add another amino acid to this would be R1, you would have the amino group on this side attached to the alpha carbon. Oopsies, kind of running off. If we form a molecule of water through a dehydration synthesis reaction, we would remove this, okay, this molecule of water, and what we form is the peptide bond. And the peptide bond would then be found between this carbonyl group and the nitrogen. So I told you if you're looking for the peptide group, you can look for con. Okay, that's how you can find that peptide group. What exactly are the functions of proteins? We said they have a very diverse, many diverse functions. They actually play tons of roles in the cell. They can play structural roles. Okay, they can provide structural support. They can be involved in cell transport. We are going to talk about the parts of the cytoskeleton that allow vesicles and other things to move through the cell, but also structures that allow for cell motility, that allow a cell to propel itself forward or to crawl forward. So there's structural, there's cell transport, there are enzymatic, and enzymes tend to play important roles in chemical reactions. They catalyze chemical reactions. Okay, so there's all of these different functions for proteins. Finally, we have the macromolecule lipids. Okay, and lipids, remember I mentioned that lipids are not considered polymers. So they don't form a repeating unit. So we don't classify them as polymers. So they don't actually technically have a monomer group. We did talk about the structure of several important lipids. We talked about triglycerides. Tri, meaning 3. Glyceride indicates that it is attached to a glycerol molecule. And triglycerides do have a special type of bond found in them. So if you look at the structure of glycerol, it's a 3-carbon alcohol. So here's the OH groups. And then you have H's on all other sides. So you notice there are 4 things attached to the carbon in every single case. This right here is glycerol. And to build a triglyceride, we take a glycerol and we attach 3 different fatty acids. Now a fatty acid has, the acid group is the carboxylic acid. And then this is attached to a hydrocarbon chain. So I'm actually not even going to finish the hydrocarbon chain, you can imagine that it would go off into the bottom here. Okay, so this has got carboxylic acid, and then there's that hydrocarbon chain going down. That's my second fatty acid. And then here is my third fatty acid. Okay, so to form a triglyceride, we do have to form a special type of bond, and that is called an ester bond. So an ester bond is used to form a triglyceride, but I want to emphasize that this is not a polymer. Okay, so this is just building a triglyceride. So to do that, we would take and do a dehydration synthesis reaction. We would take out 2 hydrogens, 1 oxygen from each of these, and we would form 3 ester bonds. Okay, so that's for lipids. What are the function of lipids? Okay, so the function for lipids, important function number 1 is energy storage. And our lipids, our triglycerides, are considered our long-term energy storage. So this is long-term. Okay. Second purpose for lipids is that they are a very important part of the cell membrane. Okay, so phospholipids make up the phospholipid bilayer. The other lipid that plays an important role in membranes is cholesterol, which is one of the sterols. Okay, and then finally with lipids, some specialized lipids serve a very interesting role in cell signaling. So you might not realize this, but there are several hormones that are actually lipids, and these include vitamin D, does function as a hormone. It is a signaling molecule. You might have heard of testosterone and estrogen. And these are also hormones that are lipids, and so they play an important role in signaling. Okay, so that is it for the different types of macromolecules.